There's a jellyfish floating in the Mediterranean right now that has been alive before you were born, before your parents were born, before their parents were born. But here's the thing that should break your brain. It didn't just survive that long. It died multiple times, and then it came back. Not metaphorically, not in some poetic sense. This creature, Turritopsis dohrnii, literally reverses its aging process, transforms back into a juvenile, and starts over, again and again, potentially forever. And that's just one organism. There are creatures living today that were alive when the pyramids were being built, trees that started growing before humans invented writing, sharks swimming through Arctic waters that were born before Shakespeare wrote a single word. These aren't myths. These aren't exaggerations. These are real organisms that have cracked codes we didn't even know existed. So here's the question that should haunt you. Why does anything die at all? Because death isn't inevitable. It's not written into the fundamental laws of physics. It's not required by thermodynamics. Death is an evolutionary strategy, a solution to specific problems, which means some organisms found different solutions. And when you look at what they did, when you actually examine how they cheated the one rule we thought was absolute, everything you thought you knew about life, about time, about the inevitability of your own mortality, changes. This is the story of the organisms that refuse to die. And more importantly, it's the story of why they refuse to die, what they know that we don't, what they've figured out that evolution decided we shouldn't have. Because if immortality is possible, and we're about to prove that it is, then every death, including ours, becomes a choice evolution made, not a law of nature. And that changes everything. Let me start with the jellyfish that breaks time itself. Turritopsis dohrnii. It's small, translucent, about 4.5 millimeters wide. You could hold a dozen of them in your palm and barely notice. There's nothing about it that screams biological impossibility. But watch what happens when it gets old, or injured, or stressed, or starved. Most jellyfish live fast and die young. They mature, they reproduce, they die. Simple. Life cycle. The life cycle we expect. But when Turritopsis reaches the end, when its body starts failing, when it should die, it doesn't. Instead, it sinks to the ocean floor. Its bell-shaped body collapses, its tentacles retract, and then, at the cellular level, something that shouldn't be possible. Its cells start changing. Not dying, not degrading, transforming. The process is called transdifferentiation, and it's one of the most bizarre phenomena in all of biology. Here's what you need to understand. Every cell in your body has a job. Muscle cells contract, nerve cells transmit signals, skin cells form barriers. Once a cell differentiates, once it becomes a specific type, that's supposed to be permanent. You can't turn a heart cell back into a stem cell. You can't transform a brain cell into a liver cell. Except Turritopsis can. When this jellyfish hits the reset button, its specialized cells, the ones that make up its muscles, its nervous system, its reproductive organs, all revert back to their embryonic state. They become stem cells again, blank slates. And then the entire organism rebuilds itself from the bottom up. The adult jellyfish essentially dissolves into a blob of undifferentiated tissue called a polyp. And from that polyp, a new juvenile jellyfish grows, genetically identical to the one that just died. Same organism, just younger. It's not reincarnation, it's not reproduction, it's transformation. The same individual, born again. Now here's where it gets truly strange. This isn't a one-time emergency escape hatch. Turritopsis can do this over and over and over. Every time it reaches the end of its life cycle, it can reverse course, theoretically, forever.
The researchers who discovered this weren't sure what they were seeing at first. This was in the 1990s. A marine biology student named Christian Summer was studying jellyfish in the lab when he noticed something impossible. The adult jellyfish he was observing didn't die. They transformed back into polyps. He thought it was contamination. He thought he'd made a mistake. He checked his tanks, his water samples, his records. Everything was clean. The jellyfish were genuinely reversing their own aging. When the paper was published, the scientific community barely noticed. It seemed too strange, too niche, too outside the realm of what mattered, a weird quirk of a tiny jellyfish nobody had heard of. But over the next decade, as more researchers studied Turritopsis, the implications started becoming clear. This wasn't just a biological curiosity. This was a fundamental challenge to everything we thought we understood about aging and death. Because here's the question nobody could answer. If this jellyfish can reverse aging, why can't we? What is Turritopsis doing at the cellular level that our cells can't do? And more importantly, why can't our cells do it? The answer involves something called cellular senescence. In most organisms, including humans, cells have a built-in limit to how many times they can divide. This limit exists because of telomeres, protective caps on the ends of chromosomes that shorten with every cell division. When telomeres get too short, the cell stops dividing. It enters senescence, a kind of retirement mode. Eventually, it dies. This is one of the primary mechanisms of aging. Your cells literally run out of runway. But Turritopsis cells don't play by these rules. During transdifferentiation, the telomeres reset. The cellular clock rewinds. The damage that accumulated over the lifetime of the organism gets erased. It's not just that the jellyfish looks younger. At the molecular level, it is younger. Now, before you think this is the secret to human immortality, let me hit you with the problem. We don't fully understand how Turritopsis does this without triggering cancer. See, in humans, cells that ignore the normal limits on division and refuse to die are called cancer cells. They're one of the things that kill us. So when you talk about reversing cellular aging, you're walking a razor-thin line between immortality and uncontrolled growth. Turritopsis somehow walks that line perfectly. Its cells de-differentiate, reset, and then redifferentiate into healthy functional tissue. No tumors, no runaway cell division, just a clean, controlled transformation. How? We still don't entirely know. Researchers have identified genes involved in the process, genes related to DNA repair, cell cycle regulation, and stem cell maintenance, but the full mechanism remains a mystery. And here's the truly fascinating part. Turritopsis isn't the only organism that can do this. There are other jellyfish species that show similar abilities. There are flatworms that can be chopped into dozens of pieces, and each piece will regenerate into a whole new worm. There are hydras, tiny freshwater creatures that show no signs of aging at all, ever. So immortality, or at least the biological potential for it, isn't a single fluke. It's a solution evolution has discovered multiple times in multiple lineages using different strategies. Which brings us back to the original question: why do we die? If jellyfish can reset their cells, if hydras can live indefinitely, if biological immortality is possible, then why don't humans have it? Why don't mammals have it? Why don't most animals have it? The answer is going to surprise you, because it's not that we can't be immortal, it's that evolution chose for us to die. And the reason why is actually brilliant. But before we get to why evolution chose death, we need to talk about the organisms that made the opposite choice, the organisms that decided immortality was worth the cost. Because there's a creature right now living in the freezing waters of the Arctic that was swimming when the printing press was invented, and it's not even close to dying. In 2016, a team of scientists pulled a Greenland shark from the icy waters near Greenland's coast. They measured it, took tissue samples, ran tests on its eye lens using radiocarbon dating, a technique that measures isotopes left over from the Cold War-era nuclear tests. When the results came back, the researchers had to double-check their calculations because according to the data, this shark was approximately 392 years old. Let me put that in perspective. This shark was born around 1624, the same year that the Dutch founded New Amsterdam, which would later become New York City. When this shark was young, the Mayflower had landed just 4 years earlier. This animal is older than the United States, older than the Industrial Revolution, older than calculus. And it's still alive, still swimming, still growing. The Greenland shark, Somniferous microcephalus, holds the record for the longest.
There's a tiny organism living in freshwater ponds and streams all over the world. It's about 10 millimeters long, looks like a small tube with tentacles on one end. You've probably never heard of it. Most people haven't. But this creature, Hydra vulgaris, might be the most important organism in the history of aging research. Because in over a decade of careful observation, studying thousands of hydras across multiple generations, scientists have documented something that shouldn't exist. These animals show zero signs of aging. Not they age slowly, not they live a really long time. They don't age, period. A hydra that's 1 year old is biologically identical to a hydra that's 10 years old. Same mortality rate, same reproductive capacity, same cellular function. The curve that represents aging in every other animal on Earth, the gradual decline, the increasing probability of death, is completely flat in hydras. They are, as far as we can tell, biologically immortal. Now, before you think this is the same as the jellyfish, let me stop you, because what hydras are doing is fundamentally different from Turritopsis. The jellyfish resefts. It transforms back to youth when it gets old. Hydras never get old in the first place. They don't need a reset button because their biological clock never starts ticking. So how is this possible? The answer lies in something that sounds almost too simple to be true: stem cells. Your body is made up of trillions of cells. Most of them are specialized: muscle cells, nerve cells, skin cells. And most of them are slowly dying. Your skin cells last a few weeks before they're replaced. Red blood cells last about 4 months. Even neurons, which live the longest, accumulate damage over time. You have stem cells that can generate new specialized cells, but even your stem cells deteriorate. They divide less frequently, they make more errors, the quality of replacement cells declines. This is aging at the cellular level. But hydras are built differently. Approximately 70% of a hydra's cells are stem cells, not differentiated tissue, not specialized cells reaching the end of their lifespan. Stem cells, constantly dividing, constantly replacing, constantly renewing. Every single cell in a hydra's body is replaced roughly every 20 days. Think about that. The entire organism is perpetually brand new. By the time a month passes, there isn't a single molecule from the old hydra remaining. It's like the ship of Theseus, but biological. If you replace every plank in a ship, is it still the same ship? If you replace every cell in an organism, is it still the same organism? Philosophically, we can debate that, but biologically, the answer is yes, because the pattern remains, the structure, the genetic code, the identity. And crucially, those stem cells never run out. They never hit the cellular limit that causes aging in other organisms. The telomeres in hydra cells don't shorten. The DNA repair mechanisms don't degrade. The quality control systems don't fail. Researchers at Pomona College studied a population of hydras for 8 years, monitoring them continuously. They tracked mortality rates, reproductive output, cellular function, and they found no statistically significant increase in mortality over time. None. The chance of a hydra dying on any given day was exactly the same whether it was 1 week old or 8 years old. That's unheard of in biology.
Every other animal shows a mortality curve that increases with age. Even the Greenland shark, despite its extreme longevity, eventually dies. Its mortality rate increases, just very, very slowly. But hydras break the pattern entirely. Now, here's where it gets even stranger. Hydras don't just avoid aging. They're almost impossible to kill through normal means. Cut a hydra in half, and both pieces will regenerate into complete organisms. Cut it into several pieces, as many as you want, and each piece, as long as it contains enough cells, will regrow into a full hydra. You can turn a hydra inside out, literally reverse its body so its digestive cavity is on the outside, and it will flip itself back and continue living like nothing happened. Blast it with radiation levels that would kill almost any other organism, and it will repair the DNA damage and survive. The regenerative capacity is so extreme that in the 1740s, when naturalist Abraham Tremblay first documented it, the scientific community didn't believe him. They thought he was lying or hallucinating. It seemed like magic. But the explanation isn't magic. It's stem cells. Relentless, immortal, endlessly renewing stem cells. And this raises a fascinating question. If stem cell renewal is the key to biological immortality, why don't all organisms do this? Why did evolution create aging in the first place, when the solution, eternal stem cell renewal, clearly exists? The answer has to do with complexity and cancer risk. Hydras are simple organisms. They have no organs, no complex tissues, no brain. Their body plan is just 2 cell layers with a hollow digestive cavity in between. This simplicity allows for constant cellular turnover without major risk. But when organisms become more complex, when they evolve brains, hearts, kidneys, specialized tissues that require precise organization, constant cellular division becomes dangerous. Because here's the problem. Every time a cell divides, there's a chance of error, a mutation, a copying mistake. Most mutations are harmless, some are fatal to the cell. But occasionally, a mutation causes the cell to ignore normal growth limits, to divide uncontrollably. That's cancer. And cancer risk increases with the number of cell divisions. More divisions, more chances for something to go wrong. So complex organisms face a trade-off. They can either maintain eternal stem cell renewal and risk constant cancer, or they can limit cell division, accept aging, and reduce cancer risk. Evolution, in most complex organisms, chose aging. Hydras can maintain unlimited stem cell renewal because their simplicity keeps cancer risk manageable. But mammals, birds, complex organisms with intricate systems? The calculation changes. Aging becomes the safer bet. But wait, if that's true, then how do some complex organisms live for centuries? How does the Greenland shark live for 500 years without dying of cancer? The answer involves something researchers call the Peto paradox. Richard Peto, a statistician and epidemiologist, noticed something strange in the 1970s. Large animals like elephants and whales should have much higher cancer rates than small mice like mouse. Why? Because they have more cells. More cells means more opportunities for mutations. More mutations means more cancer. But in reality, elephants and whales don't get cancer more often than mice. In fact, they get it less. This shouldn't make sense. But it does once you understand what these long-lived animals evolved. Elephants, for example, have extra copies of a gene called TP53, a tumor suppressor gene. Humans have 1 copy. Elephants have 20. These extra copies mean that when a cell starts showing signs of becoming cancerous, it's detected and destroyed far more efficiently. Whales evolved even more sophisticated DNA repair mechanisms. Their cells are extraordinarily good at fixing mistakes before they become cancer. Greenland sharks likely have similar adaptations, though we're still figuring out the details. Their slow metabolism helps. Fewer cell divisions per year means fewer chances for cancer. But they also appear to have enhanced DNA repair and cellular quality control. In other words, some animals can live for centuries without cancer, but only because they evolved extremely expensive defense systems, systems that require genetic real estate, metabolic energy, and developmental complexity. For most species, it's not worth the cost. Better to age and die than to invest that much into cancer prevention. But hydras bypass the whole problem by staying simple. And that's the key insight here. Immortality isn't impossible, it's just expensive. And for most organisms, death is cheaper. But hydras aren't the only organisms that chose simplicity and immortality. There's another creature, even stranger, that makes hydras look fragile by comparison. Because there are organisms living today that can survive being boiled, frozen to near absolute zero, blasted with radiation 1,000 times stronger than what would kill you instantly, and even launched into the vacuum of space. And when conditions improve, they just wake up. Meet the tardigrade. Also called water bears or moss piglets, these microscopic animals are about 1/2 millimeter long. They have 8 legs, a segmented body, and a kind of lumbering gait that makes them look like tiny, chubby bears under a microscope. They're adorable, almost cartoon-like, and they are the most indestructible complex organisms on Earth. I'm not exaggerating. Let me list what tardigrades have survived in laboratory conditions. Temperatures as low as -272 degrees Celsius. That's 1 degree above absolute zero, the point where atomic motion nearly stops. Temperatures as high as 151 degrees Celsius. Boiling water is 100 degrees Celsius. Tardigrades laugh at boiling water.
In the White Mountains of California, at an elevation where almost nothing else can survive, there's a tree that was already ancient when Rome fell. It's a bristlecone pine, twisted, gnarled, barely alive. Most of its trunk is dead wood, bleached white by thousands of years of sun and wind. But there's a thin strip of living bark, maybe just a few inches wide, still connecting roots to a handful of branches that still produce needles. This tree is 5,067 years old. Let me put that in perspective. When this tree first sprouted from a seed, humans had just invented writing. The wheel was brand new technology. The pyramids of Egypt hadn't been built yet. This tree is older than recorded history. And it's not even the oldest. There are bristlecone pines in those mountains that are over 5,000 years old. And in Sweden, there's a Norway spruce whose root system has been alive for 9,550 years. That tree started growing at the end of the last ice age. 9,500 years. Think about everything that's happened in human history. All of it, every empire, every war, every discovery, every invention. That tree has been alive the entire time. So how is this possible? What allows a tree to live for millennia? The answer starts with something we've already discussed, but it works differently in plants. Growth. Continuous, indefinite growth. Animals have what's called determinate growth. We grow until we reach adult size, then we stop. A human doesn't keep getting taller throughout their entire life. A dog doesn't keep growing until it dies. We reach a genetically programmed size, and that's it. But plants have indeterminate growth. They never stop growing. As long as conditions allow, they keep adding new tissue, new branches, new roots, new leaves. And here's the key. That new tissue contains new cells, young cells, cells that haven't accumulated damage. Trees grow from meristems, regions of actively dividing stem cells located at the tips of branches and roots. Every year, these meristems produce new wood, new bark, new growth, and these meristematic cells never age. Think about it. The cells at the growing tip of a 5,000-year-old bristlecone pine are just as young as the cells that formed when the tree first sprouted. The tree doesn't age because it's constantly producing new, undamaged tissue. The old wood, it dies. It becomes structural support, dead tissue holding up the living parts.
But the living tissue itself, the thin layer of cells just beneath the bark, called the cambium, stays perpetually young. It's like if humans could grow new organs every year and let the old ones die off while keeping the new ones functioning. We'd never age either. But there's more to it than just continuous growth. Bristlecone pines survive for millennia because they live in one of the harshest environments on Earth. They grow at elevations between 9,000 and 11,000 feet in the White Mountains. The soil is poor, mostly limestone and dolomite. Water is scarce. The growing season is maybe 6 weeks out of the year. Winter temperatures can drop to -40 degrees. Summer brings intense UV radiation and desiccating winds. It's a terrible place to be a tree, which is exactly why they live so long. In harsh environments, trees grow slowly. A bristlecone pine might add only 1/100th of an inch to its radius in a year. Some years, in particularly harsh conditions, they don't grow at all. Their growth rings are so tightly packed, you need a microscope to count them. Slow growth means dense wood. Dense wood is more resistant to rot, to insects, to fire, to all the things that kill trees. And slow growth means slow metabolism, less cellular division, less accumulated damage. The trees are also chemically defended. Their resin has antimicrobial properties that prevent fungal infections and insect infestations. Even after sections of the tree die, the wood doesn't rot. It just stands there, weathered into smooth, sculptural forms, but structurally intact. There are bristlecone pines in the White Mountains that are half dead, 3/4 dead. Some are 95% dead wood with just a thin ribbon of living tissue keeping a single branch alive. And they persist like that for centuries. It's a kind of immortality through minimalism. The tree sheds everything non-essential, reduces its living tissue to the bare minimum, and that minimum just keeps going. But here's what's truly strange about tree longevity. The oldest trees aren't always the largest trees. In fact, some of the oldest bristlecone pines are relatively small, stunted by harsh conditions. Compare that to coast redwoods or giant sequoias. These trees can grow to enormous sizes, some over 300 feet tall, but they rarely live beyond 3,000 years. They grow fast in favorable conditions, reach enormous size, and then succumb to fire, disease, or structural failure. The bristlecone pines live longer precisely because they grow slower, stay smaller, and exist in environments where competition is minimal. It's the tortoise and the hare, but on a millennial timescale. And this brings us to a pattern we've been seeing throughout this entire journey. The organisms that live the longest aren't the strongest, the fastest, or the most competitive. They're the patient ones, the efficient ones, the ones that minimize risk and maximize stability. Greenland sharks live in cold, deep water and move slowly. Hydras are simple and renew constantly. Tardigrades shut down entirely when conditions turn bad. Bristlecone pines live in harsh places where nothing else can compete with them. Immortality, or near immortality, isn't about conquering your environment. It's about finding a niche where you can persist with minimal interference. But this raises the question we've been building toward this entire time. If biological immortality is possible, if organisms can live for centuries or indefinitely, if aging can be avoided through various strategies, then why do most organisms, including humans, age at all? Why did evolution create death? Because make no mistake, aging isn't an accident. It's not a flaw in the system. It's a deliberate evolutionary strategy. And when you understand why evolution chose aging for most complex organisms, everything else starts to make sense. The answer is going to challenge everything you thought you knew about life and death. Here's the thing that breaks most people's brains when they first learn it. Aging didn't evolve because it's good for the individual. It evolved because it's good for the species. Let me explain what that means. Imagine two populations of organisms. Population A is biologically immortal. They don't age, they just keep living and reproducing indefinitely as long as nothing kills them. Population B ages normally. They reproduce, they get old, they die. At first glance, you'd think population A has an advantage, more time to reproduce, more opportunities to pass on their genes. Immortality seems like an obvious evolutionary win. But there's a problem, and the problem is resources. In any environment, there are limited resources: food, water, space, whatever the organism needs to survive. If individuals never die, the population keeps growing. Eventually, you hit carrying capacity. The environment can't support any more individuals. Now what happens? The immortals keep living. They keep consuming resources, but they're also occupying all the available space and consuming all the available resources. There's no room for new generations, no opportunity for young individuals to establish themselves. And here's the critical part. If the environment changes, if new challenges arise, the immortal population can't adapt quickly. Evolution works through variation in offspring, new mutations, new combinations of genes. If you have a population of immortals that reproduce slowly and never die, you have very little variation, very little raw material for natural selection to work with. But population B, the aging population, they're constantly cycling. Old individuals die, new individuals with new genetic combinations are born. When the environment changes, this population has variation. It can adapt, it can evolve. Over the long run, across thousands or millions of years, the aging population outcompetes the immortal population because it's more flexible, more adaptable, better able to respond to change. This is called the theory of programmed aging, or the adaptive aging theory, and it's one of several explanations for why aging exists. But there's another theory, perhaps more widely accepted, called the disposable soma theory. It argues something slightly different but equally important. Every organism has limited energy, limited resources. You can invest that energy in maintenance and repair, keeping your body pristine indefinitely. Or you can invest it in reproduction, in making as many offspring as possible before something kills you. But here's the reality of nature. Most animals don't die of old age. They get eaten, they starve.
In a laboratory in San Francisco, a team of researchers just did something that should be impossible. They took skin cells from a 114-year-old woman and reprogrammed them back into stem cells. Then they grew those stem cells into neurons, heart cells, blood cells, young cells, functionally identical to cells from a newborn. They reversed aging, not in an entire organism, but in cells. They took the oldest human cells ever studied and made them young again. The technique is called cellular reprogramming, and it works by activating a specific set of genes called Yamanaka factors, named after Shinya Yamanaka, who won the Nobel Prize for discovering them in 2012. These 4 genes can take a specialized adult cell and essentially rewind it back to an embryonic state. It's the closest thing we have to what the immortal jellyfish does naturally, and we can do it in a lab, reliably, repeatedly. Now here's where it gets interesting. If we can reverse aging in cells, why can't we reverse aging in an entire organism? Why can't we make a 114-year-old young again? The answer is the same problem we keep running into: cancer. When you activate Yamanaka factors in an entire living organism, you don't just reverse aging, you also destroy cellular identity. Neurons forget their neurons, liver cells forget their liver cells. Everything reverts to stem cells, and the organism falls apart. But in 2016, researchers at the Salk Institute tried something clever. Instead of activating the Yamanaka factors continuously, they pulsed them. Short bursts, just enough to partially reverse aging without completely erasing cellular identity. And it worked. They tested it on mice with a premature aging disease.
The mice treated with pulsed Yamanaka factors lived 30% longer than untreated mice. Their tissues showed signs of rejuvenation. Their organs functioned better. They didn't just live longer, they lived younger. This is called partial reprogramming, and it's one of the most promising approaches to reversing aging currently being researched. The idea is to dial back the aging clock without completely resetting it, to make cells younger without destroying what makes them specialized and functional. And it's not just happening in mice. There are biotech companies right now, funded by billions of dollars, working to bring this technology to humans. Altos Labs, founded in 2021, has raised over $3 billion to research cellular rejuvenation. Calico, backed by Google, is investigating the biology of aging and longevity. Unity Biotechnology is developing drugs to clear senescent cells, the zombie cells that accumulate in aging tissues and cause inflammation and dysfunction. The science is moving faster than most people realize. We're not talking about science fiction anymore. We're talking about clinical trials, about therapies that might be available within our lifetimes. But cellular reprogramming is just one approach. There are dozens of other strategies being explored right now, each targeting different aspects of the aging process. There's senolytics, drugs that selectively destroy senescent cells. Early trials in humans have shown promising results. People treated with senolytics report improvements in physical function, reductions in inflammation, even improvements in age-related diseases. There's NAD+ boosters, compounds that increase levels of a molecule critical for cellular energy production and DNA repair. NAD+ levels decline with age, and restoring them in mice improves metabolism, enhances DNA repair, and extends lifespan. Human trials are ongoing. There's rapamycin, a drug that inhibits a cellular pathway called mTOR. Reducing mTOR activity extends lifespan in every organism it's been tested in, from yeast to mice. Some longevity researchers are already taking it themselves, though long-term effects in humans are still being studied. There's metformin, a diabetes drug that appears to have anti-aging properties. People who take metformin for diabetes live longer than people who don't have diabetes at all, which shouldn't make sense unless metformin is doing something beyond just controlling blood sugar. And then there's genetic engineering. Scientists have already identified genes that, when modified, extend lifespan in model organisms. The record holder is a nematode worm modified to live 10 times longer than normal. 10 times. Imagine if humans could live to 700 or 800 years with a similar modification. We're nowhere near doing that in humans, obviously, but the principle is proven. Lifespan is genetically malleable. It can be extended through intervention. And then there's the most radical approach of all, not extending life, but pausing it. Human cryopreservation. The idea is to preserve the body, particularly the brain, at extremely low temperatures immediately after death. The hope is that future technology will be able to reverse the damage, repair the tissues, and revive the person. It sounds like science fiction, and maybe it is. The technology to revive a frozen human doesn't exist yet, but the technology to preserve one does. There are people frozen right now, stored in liquid nitrogen, waiting for a future that may never come. But here's the thing that should make you think. We can already freeze and revive simple organisms. Tardigrades do it naturally. We can freeze human embryos and revive them decades later. We've successfully frozen and revived rabbit kidneys, though not human organs yet. The principle works. The question is just how much complexity we can preserve and recover. Can we preserve the intricate neural connections that encode memory and personality? Can we reverse the damage caused by ice crystal formation? Can we revive someone who's been legally dead? Nobody knows, but there are researchers working on it, and every year the technology improves. So let's say hypothetically that one or more of these approaches works. Let's say that within the next 50 years, We develop therapies that can significantly extend human lifespan, not just to 120 or 150, but to 200, 500, maybe indefinitely. What then? What happens to humanity if we defeat death? Because this isn't just a medical question, it's a civilizational question, and the answer isn't as simple as you might think. Let's start with the obvious problems. Overpopulation. If people stop dying, or even just die much more slowly, and they keep having children, the population will explode. Earth already has 8 billion people. What happens when that becomes 16 billion, 50 billion? When does the planet run out of resources? The counterargument is that birth rates decline as societies develop. Most developed nations already have birth rates below replacement level. If people live indefinitely, they might choose to have fewer children or no children at all. The population might stabilize. But that's just one issue. What about inequality? Right now, aging is the great equalizer. Rich or poor, everyone gets old, everyone dies. But if anti-aging treatments are expensive, and they almost certainly will be at first, then only the wealthy will have access. The rich will live for centuries, while the poor age and die normally. You'd have a society divided not just by wealth, but by time. A permanent upper class that accumulates centuries of experience, knowledge, and power, while everyone else lives normal lifespans. That's not just inequality. That's a caste system based on mortality. And what about the psychological effects? Humans aren't built for immortality. Our brains evolved in a world where death was always near. Our sense of urgency, our motivation, our appreciation for life, all of it is shaped by the knowledge that time is limited. What happens when time stops being limited? Do people lose their drive? Do they become complacent, bored, depressed? There's a philosophical thought experiment called the Tiphanos problem, named after a figure in Greek mythology. Tiphanos was granted immortality, but not eternal youth. He aged forever, becoming more and more decrepit, unable to die. Eventually, he begged for death, but it never came. The lesson is clear. Immortality without quality of life is torture. If we extend lifespan, we have to extend health span too. Living to 500 means nothing if you spend 400 of those years in pain, disabled, dependent on others. But let's say we solve all of that. Let's say we can extend lifespan and health span. Let's say we create a world where people can live indefinitely in good health, where the treatments are accessible to everyone, where we've solved the resource and population problems. Would you want to live forever? Because here's the thing that nobody talks about. Immortality means watching everyone you love die. Unless they're also immortal, you'll outlive your friends, your family, your children. You'll form relationships knowing they're temporary. You'll experience loss over and over again, and over again. And even if everyone is immortal, relationships change. Marriages that last 50 years are considered successful now. What about marriages that last 500 years? 5,000 years? Do people stay together, or do relationships become temporary by